Hello friends, today we are going to discuss a new topic, bacteriocins. The bacteriocins are one of the major concern in the food industry because of the contamination of the food by pathogens, which are the frequent cause of the foodborne diseases. Over the past decade, the recurrent outbreak of the diarrhea combined with the natural resistance in such causative agents has contributed to its status as hazard. So there was a demand for safe food with less chemical additives, and there was a great interest in replacing this compound by natural products which do not injure the host or the environment. Means it does not cause uh, any kind of injury to the host and the environment. So there was a possibility to find a solution which was natural as well as safe. So the use of non-pathogenic microorganisms and their metabolites to improve the microbiological safety and the extent of shelf life of the food. We call it as biopreservation, and this idea of biopreservation uh, brought about a revolution to bacteriocins. Now, we should know what bacteriocins are. See, these bacteriocins, they are the proteinaceous toxins which are produced by bacteria, and they comprise of a huge family of ribosomally synthesized peptides, which has the properties such as they are colorless, they are odorless, they are tasteless. Now, these bacteriocins, they have antimicrobial activity against either the similar or the closely related bacterial strains. They were discovered by Gracia in the year 1925, who was finding out a way to kill the bacteria in a natural way. So these bacteriocins he found to have the antibacterial activity against the closely related bacterial strains. Now, if we talk about the types, their classification, because as the word of bacteria consists of millions and trillions, even we can say countless of bacteria, so they can be classified based on different bases. First classification basis is the producing strain, means what kind of microorganisms are producing them. So based on that, we have different type of bacteriocins. What kind of resistance mechanisms are developed by the host against bacteriocins is another classification basis. What is the killing mechanism? How is their molecular weight? Some are large molecular weight peptides, some are smaller, some are medium. Based on that, they can also be classified. And the last is their genetics, their structure and their function. How do they work? We have four different classes: class 1, 2, 3, 4. These different classes are included in the last category. So we'll take up each of these one by one. Now, the first type of basis of classification is the production strain, which actually produces the bacteria. Based on the production strain, the first type are the colicins and the microcins. Colicins, as you may guess, are produced by E. coli, and we all know E. coli is a gram-negative bacteria which belongs to Enterobacteriaceae family. However, the colicins and microcins, they are different in their size. Their production trigger means the factor which are going to stimulate their production, and their release mechanism. But they both are produced by the members of Enterobacteriaceae family. The second, we have the archaeocins. They are produced by the extremophiles, or we can say the bacteria living in the extreme environment. And the example include sulfolobicin, which is uh, which is found in the hot acidic environment. In addition to this, we yet have another example, that is halocins, which is a salt-loving bacteria which grow in the high saline environment. Now, the colicins and the microcins, both they target the cellular functions like the membrane disruption and protein synthesis inhibition in the host. Means they both are going to cause the same kind of uh, effect. They both are going to cause the same kind of attack on the target. However, there is a little bit difference that the colicin, they can form the pores on the membranes, and they can degrade the peptidoglycan precursor, means which is the molecule which is required for the synthesis of peptidoglycan, which protects the gram negative bacteria. And the colicin also requires some specific outer membrane receptor for its entry into the cell. However, if I talk about the microcins, they are yet smaller than colicins, and they also form pores, but they target the ATP synthetase, which is also required for the synthesis of ATP. Now, if I talk about the second basis of classification, that is common resistance mechanism, which involves the target modification, active efflux, enzymatic activation, and immunity proteins. Among them, the target modification involves, means they are going to modify the target cell in the cell envelope. They can modify the cell envelope, or they can inactivate the target, means they are going to inactivate the target cell. So uh, the the bacteriocin is going to get modified by the target cell. Active efflux, it includes the bacteria which use the transporter system to pump the bacteriocins out of the cell, and they prevent the accumulation, means the bacteriocins won't be able to accumulate inside the host, so that its effect is minimized. The third is the enzymatic inactivation, where the specific proteases, they cleave or they inactivate the specific bacteriocins, uh, as it happens similar to the case of antibiotic resistance. The last is the immunity protein, where the bacteriocin producers, they have specific immunity protein. They bind to the bacteriocin, forming a complex that blocks its action, or they export it. So these are the different mechanisms which gives resistance against the bacteriocins in the host. So that is also one of the criteria for classifying the bacteriocins. The third is the killing mechanism. How are they going to cause the death of the host cell? The common resistance mechanisms were those which were acquired by the host. Now, killing mechanisms are those which are acquired by the bacteriocins, which are possessed by the bacteriocin in order to kill the host cell. Now, among the different killing mechanisms, the first one is the membrane disruption. The bacteriocins like lacticin, one of the examples is there, which is a cationic bacteriocin that get inserted into the negatively charged bacterial membrane, and it forms channels or pores, um, thereby losing the cell loses its integrity and the cell dies. The second one is inhibition of biosynthesis, which includes the inhibition of the cell wall synthesis. Um, See, cell wall is one of the very important component which maintains the integrity of the cell. And if the cell wall synthesis is inhibited, the cell is going to die. So, the inhibition of cell wall synthesis may be either by targeting the internal cellular processes, which are conducted by the enzyme. The third killing mechanism may be interference with the cellular processes, and the cellular processes like signaling, the enzyme modulation, they all can be there. And if these cellular processes they are disrupted, they are halted, the cell won't be able to survive. And the last one is cell lysis, where the bacteriocins are going to cause the bursting of the cell or the lysis of the cell. So these were the different killing mechanism, how the bacteriocins are going to kill the host cell. Now, the fourth criteria, the fourth basis for classification is the molecular weight. The bacteriocins are of different molecular weight. They are uh, they are of different sizes. So based on that, they are of four different type. It could be a very small peptide, we can say less than 10 kilodalton. And the example is plantaricin, plantaricin, sorry. And these plantaricins are 1.3 kilodalton peptides. And the second category is the medium ones, medium bacteriocins, and bacteriocin ZFM 216 is the example, which has around 11.8 kilodalton molecular weight. The third category is of colicins, which have the molecular weight of around 30 to 80 kilodaltons, and they are produced from E. coli. We know them well. The fourth category are the large bacteriocins, which has a molecular weight of more than 80 kilodalton, and the the phage-like proteins are the examples of this high molecular weight or large molecular weight bacteriocins. See, the size influences that how they interact with the bacterial membrane, means the pore formation, the cell wall inhibition, how all these mechanisms are going to work, that depends on the size of the bacteriocin. Now, the last basis for the classification of bacteriocins involve the class 1, class 2, class 3, and class 4. Taking each individually, if I talk, the class 1 bacteriocins, they are lentibiotics, right? And these lentibiotics, they are they undergo the extensive post-translational modifications. Means they have some unusual amino acids like the lanthionine or the 3-methyl-lanthionine included in it, due to which it is called a lentibiotic, not an antibiotic, but it is a lentibiotic because of the modification, post-translation modification, which modifies an amino acid to lanthionine. Such modification may lead to complex, stable structures, and one of the very well-known example is niacin, which was the first first bacteriocin to be discovered. The class 2 bacteriocins, they lack the extensive post-translational modification, and they form the amphipathic helical, or we can say the beta sheet structures in the target membrane, and sometimes they contain the disulfide bond for their stability. They are relatively small. They are heat labile, means they get degraded by the action of the heat, and plantaricin is the example. If I talk about the third category, which are the large protein, or I can say they are more than 30 kilodalton. They are heat labile again, and bacteriolysine is the example, and it degrade in the target cell wall. The last category in this is the class fourth, bacteriocins, which are the complex bacteriocin. Sometimes they are found in circular structures, or they have a unique head to tail cyclized backbone. And because of their cyclic structure, they have the exceptional stability. The cyclic structure may be formed because of the bonding between the lipids and the carbohydrate components within them. Now, moving on to the discussion that why should we prefer the bacteriocins over the conventional antibiotics? Why are we more interested in the bacteriocins rather than the conventional antibiotics? So we all know that they are made from non-pathogenic bacteria. So there are less chances that they are going to be dangerous for us. They will be they will be in our favor. Moreover, um, they provide us the interesting group of biomolecule with the antimicrobial properties. Now, they are primary metabolite in nature. This means that bacteriocins have a relatively simple biosynthetic mechanisms. Means they are synthesized from a simple mechanism as compared to the conventional antibiotics, which are actually the secondary metabolites, which have a different pathway of their formation. So, as they are easily amenable through bioengineering to increase their activity, or we can say specificity towards the target microbe. Now, owing to this feature, the bacteriocins um have become less damaging to the natural gut microflora, which is a common drawback of the conventional antibiotic use. As we all know that whenever we are supposed to take antibiotics, they are to be followed with the the probiotics. So these bacteriocins are themselves probiotics. So we need not to follow the probiotics along with them. Now, they are generally regarded as safe, means they are safe to be consumed, they are not harmful for us. So they can be an easy solution for they can be an easy solution to be used as the preservative in the food items. Now, if I talk about the mechanism of their action, that how does they work, a general sort of mechanism of their action we are going to discuss here. An example, nisin we have taken. So the bacteriocin nisin is produced by the lactic acid bacteria. Generally, we call it as, in short, lab. The the lab is the bacteriocin is produced nisin here, and it targets the bacterial cell membrane. Suppose listeria monocytogenes the bacteria is about which we are talking here. This is the listeria monocytogenes, and the nisin, it binds to the cell membrane, or in two different ways. Here you can see that either it is binding to lipid 2, and by binding to lipid 2, it causes the pore formation in the membrane, cytoplasmic membrane, due to which there is leakage of ions like sodium, potassium, ATP, and other metabolites are loosed from inside to the outside of the cell. So there is a loss of the proton motive force, we can say. Now, when this will happen, obviously the cell will not be able to survive anymore. The second way is the inhibition of the cell wall synthesis, where nisin is going to bind to the lipid 2, which is shown here on the cell wall of the target cell, and it leads to either the cell death or the growth inhibition. See, cell wall, if it is not synthesized properly, that will also lead to the growth inhibition. So this is how the bacteriocins are going to work. This is the common mechanism of their action. Now, if I talk about from where are we going to get these bacteriocins, so the simplest and the easiest source are the lactic acid bacteria. Just now we have discussed of lab. They are used as the potential preservative and therapeutic antibiotics because the lab bacteriocins, they have some special features, they have some extraordinary uh, features due to which we prefer to use them. So if I talk about the lab bacteriocins, their advantages, so they are tolerant to high temperatures, means at high temperatures also they are in their working state. Otherwise, proteins or peptides, they denature at high temperature. But lab bacteriocins produced by lab, they are tolerant to high temperatures. They are active over a wide range of pH. They work faster, and they are easily degraded by the proteolytic enzyme, due to which they do not live long in the host or in the environment. That is the important thing, uh, which we can say is a factor which due to which there is no resistance development against the bacteriocins yet. So despite the long history of the bacteriocin use, we can say there have been no reports on the development of resistance against bacteria, the reason we have in front of us. And the bacteriocin fragments do not live long, and it minimizes the opportunity of the target strain to interact with the degraded fragment, and the which is actually the starting point for the development of the resistance. So that is why we are more concerned about the bacteriocins from the lactic acid bacteria. Now, the first bacteriocin produced was nisin, as we discussed earlier. It was also obtained from lab, lactic acid bacteria, and that the genus was Lactis, Lactobacillus lactis. They are the gram-positive cocci, bacilli, or rod-shaped. They are non-aerobic, but they are aerotolerant, means they are able to survive in presence of oxygen. They ferment the carbohydrates, produce energy. In addition to energy, because the fermentation process is going on, they produce lactic acid. This nisin, which is a bacteriocin, has widely been used in cheese, sauces, and canned foods for its preservation. And in addition to the advantage of preservation, these bacteriocins, these lactic acid bacteria especially, they produce small organic molecule which can add aroma and specific organoleptic quality to the product, means which makes it more healthier, tastier, and even add flavor to the aroma to the food. So they are considered to be a good alternative. Now, during the sugar fermentation, the lactic acid is produced, which reduces the pH, and the other organic acids which are produced, they together they lead to the reduction in pH, and which is supposed to be an important factor for the inhibition of the undesired bacteria. Whatever we don't want to grow there, so they all will be inhibited because they are not able to withstand the low pH. So the fermentation process producing the lactic acid and organic acid, thereby reducing the pH, and we can say is a kind of inhibition factor for the undesired microbes. So altogether, nisin has an important role in preservation of food and the fermented products. In addition to this, it adds the organoleptic flavor to our food. Now, There have been other bacteriocins in addition to nisin, which are produced by different bacterial strains like pediosin, cytolysin, subtilicin, lacticin, and enterocin, which are produced by the respective parent bacterial strain. So these examples are important to remember. With this, we have discussed about bacteriocins, how do they work, their different types, and we'll come up with a new topic in our next lecture. Thank you so much.